Hello, I'm Ted Kobfleisch, the Chief Science and Technology Officer of Intrepid Bioinformatics, and I'm going to demo our system today, some of its functionality with respect to SNP genotype data and the raw sequence data from which it would have been derived. Our system is a data repository specifically for genetic variation data. We're very well set up for SNP and genotype data and the sequence data from which it's derived. Going to log into our secure repository. What we have in the background is an Oracle database within which we've built our security model. All data access requires username and password login. The data within the system is protected via view-based security, which means that when a record is uploaded by a user, they are the only ones who can see the record. Subsequent to that, they can securely share the data with other individuals, groups of individuals, larger consortia, up to and including the public at large. We've built the database, the data security model within the database for one specific reason, and that is that irrespective of how it is that you're accessing the data, it's impossible to circumvent the security model. So whether you're accessing it through our user interface, through our SOAP-based web services, or through direct database access, either through SQL command line or JDBC, you will not be able to circumvent our security model. So users would access our system in the same way that they would access genomic information through virtually any genome browser. You can search by gene symbol or entree ID, and what will be pulled back are a list of gene loci that are consistent with the search term, in this case the prion protein locus. We're going to browse to the human version of this gene as mapped to human assembly 36.3. And what comes up immediately is a browser window, the sort of thing that we're used to looking at. And annotated across the top are the positions that have been reported as variants, either in dbSNP or by other users who have selected to share their variant information with me. Below that, you see individual tracks for all of the transcripts that have been identified in this region. And for this particular gene, we have 6 different splice isoforms that have been identified. We can scroll down, and below this we see a tabular view of the information that's presented in the cartoon above. All polymorphisms, transcripts, and repeat regions, if they've been annotated, would show up in this view. For the polymorphisms, what we can see are those polymorphisms that have genotype data within our system. For example, this particular polymorphism has genotypes loaded for 446 samples, and those samples are distributed across these subpopulations. You can mouse over any of these bars and see what the allele frequencies are for the total or for each of the individual populations. You can click on each individual population and be taken directly to the genotype data for the samples that have been stored. And we can see these first 3 individuals I know to be related as mother, daughter, father, and we can see that the genotypes for these individuals are consistent with Mendelian inheritance, and that's something we can feel good about. We can come back to our browser and elect to search in another fashion. For example, we can search either via individual polymorphism ID. We can search by individual. The data, once it's been submitted to us, is broken down into individual genotypes and stored as an individual record within our table that can that has its own security access information associated with it. But what that gives us the ability to do is to present this information to the user or allow them to search it in really any manner that they deem necessary. So it's possible, as in this case, to search by a polymorphism across an entire population. Or if you want to search by individual or group of individuals across an entire panel of polymorphisms, that type of search is available as well. So for this polymorphism, we can see just how much genotype data we have for this particular SNP. The allele frequencies are pretty evenly broken up across all of the populations, and we can search this information in greater depth if we wish to by simply clicking on one of these bars, and we can see our trio again, daughter, mother, father. But when we look at the genotypes, we see something that's a little unsettling, and that is that the genotypes that we see here don't obey Mendelian inheritance. If the father is truly homozygous and the mother for C and the mother is homozygous for T, the daughter should be heterozygous CT. There's enough additional information on this trio that we are certain that this is the nature of their relationship, so something simply must be wrong with the genotyping call. We can probe in greater detail if we wish by simply clicking there, and what we can see is the call that was made on the Illumina SNP chip as a homozygous C, but what's also available to us is companion sequencing data from the 1,000 Genomes Project. This is a whole genome sequence data set, and what we were able to do is immediately go to the position in this map data set that corresponds to this polymorphism and pull down the alleles that map to this position in that mapped data set. And what we see is that there are 25 C's and 11 T's that are mapped to that corresponding position, which supports a heterozygous genotype call for this individual disposition, and not the homozygous CC that was made on the SNP chip. And this is valuable because we can see that if this father is in fact heterozygous at this position, then it is possible for the daughter to be homozygous T at that position because there was an inheritable T allele. That feels good and makes for a more compelling story. But if we want to go and investigate this in greater detail to see perhaps why the genotyping assay might have been compromised, we can do that. By clicking in on View in IGV, what we do is go to get the integrated genome viewer, which was developed at the Broad and the Whitehead, and we have modified it such that it will pull data securely out of our data management system. And specifically, it will go to that location in this individual's genome and download the reads, the subset of information for the reads that span this particular position, and we can load that. So we sped up that upload for the purpose of the demonstration, but on a hardwired Ethernet cable, this upload of the 25 or so thousand reads that are found in this region typically takes about 15 or so seconds. What we can see at this position is that the individual is in fact heterozygous CT with high quality base calls there. But what's more informative in terms of or with respect to the assay that would have been on the SNP chip is that on the same chromosome as the T allele that was missed by the assay, we can see 5' of about 3 base pairs 5' of that, another T allele which differs from the reference sequence, and another 10 bases upstream of that, we see a C allele that differs from the reference. And if the assay was designed against that region of the chromosome, then clearly it is not going to be able to anneal to that locus in order to pull down that T allele, and thus the only alleles that would be visible for this individual would be the C allele, which is on a chromosome that is consistent with the oligonucleotide that would have been designed to assay it. Hence, we get allelic dropout, and what's visible for the father is only the homozygous C, when in fact the correct call is heterozygous CT. So that's a quick run through of our functionality with respect to genotypes and the sequence data from which it would have been derived. Obviously, if you have any interest, please contact us, and we would love to hear anything that you have to say in terms of comments or address any questions or concerns that you might have. Thank you very much for your time.